Okay, so we're going to talk about Sol Strand 1, which is basically just the scientific method. And so just to start and refamiliarize ourselves, let's remember the acronym OHEC. Observe, sorry, my handwriting is terrible. Observe, hypothesize, experiment, and then conclude. Those are the steps, and if we follow those, we should be good. Okay, so question number 1. Does deal with science, not so much the method, but it does deal with science. So if a student needs to do research on heredity, which is basically genetics, for a science project, which of the following sources is the most reliable? So the key word here is science. When dealing with science, only one of these really works. And so you'll notice two things here. There's news, news, news. And then there's journal. So what's the difference? We have news and we have journal. So let's get into it. Now, when you're looking at news, news is money driven. They want to sell ratings. They need to have good ratings so that their advertisers who make commercials pay them well. To get the money, they need to be sensational. So a sensational bias is when you tend to report things that are more interesting. And what's the most interesting thing? Negative things. So they tend to have a negativity bias. They report things in the most negative light that they can to sell more newspapers or create more views on their channel. All right, and then controlled by editors. So Fox News, everybody knows that if you're listening to Fox News, you are probably Republican. They tend to report things from the Republican point of view. Their editors like that. And then CNN tends to be more democratic. This is just something everybody knows at this point. So you have to be aware of the agenda behind what they say. And because of all of that, it's not really scientific. Science is empirically based. You tend to just look at evidence and you ignore biases. And it's not really reviewed that much. I mean, other news stations will fight with other news stations and talk about which one is better, but in general, a story that is misrepresented is not often fixed. So a great example of that would be tabloids. So tabloids are just funny at this point. Nobody really believes them. But more relevantly, on actual news stations, we have people like Jenny McCarthy who come on with a very sensational story about how vaccines cause autism. She had a child with autism, and right before the diagnosis, she actually had a vaccine for her child. Now, this vaccine did not cause autism, but she correlated the two and assumed. And that assumption, when she went on the news, she reported that assumption led to other people not getting their child vaccinated, and those children died of preventable diseases. So she went on the news with a sensational story, and the news reported it, and they didn't really check to verify her information. So this is another reason why you should not get your science from news. So then where do you get your science from? If there's something scientific, I want you to go to a journal. So the best way to do that is just Google Scholar. They have a lot of journals that are very easily accessible, and they're free, or at least the search for them is free. Sometimes you have to pay for them. Here's why you want a journal. They must maintain integrity. If they do not, they lose scientific weight and people will not trust them. They will not use them. So to stay relevant, they have to have integrity. They have to be peer reviewed. So most of the articles there are reviewed by teams of scientists who scrutinize anything that is published to try to find it to be wrong. We are our worst critics. Scientists are very mean to each other. We try to disprove each other. We don't try to prove each other's hypotheses. We try to disprove them. We try to discredit. And if somebody's hypothesis and conclusion can withstand the scrutiny of that, then they are probably correct. All right, so then it is published by experts in the field. The people who actually publish your work have knowledge of it and can see if you're just full of crap or if you're actually got something good. So cited work as well. We tend to give each other credit for the information that we use. So if you want to further your research, you can do so by tracking down information in the work cited. And so, for example, if we were to look at that same story about vaccines, we could just use Google Scholar, do a quick search, and if you're curious of what that would find, here you go. Vaccines do not cause autism by the Clinical Vaccine Safety Resource Guide journal, and that's in Springer. That's a pretty good journal, so we can probably trust it. And there you have it. You could read on and on and on about it if you wanted to. So journals are the way to go with science, not news. So that's the answer. Sorry about the announcements. Yeah, sorry about that. I'll try to pause this when announcements come on, but I have to record it in school just because of internet at home being an issue. All right, so moving on. Number 2. So this is about microscopes. A student wants to view cells under the compound microscope at a total magnification of 400x. If the eyepiece is 10x, which of the following objective lenses should be used? So for microscopes, there are 2 lenses. This is the first lens here, that's called the ocular. And then these lenses here, which are adjustable, are called the objective. And it's I'm sorry for my handwriting. I'm trying to write on a tablet while I'm standing. So it's very simple. It's just ocular times objective equals the total. And in this case, the total that they're looking for is 400. And they're saying that the eyepiece, which is the ocular, this thing here, the eyepiece is 10. So 10 times what equals 400? That's right, 40. So just to be clear here, this is 10. And this one right here, the red one, is actually, sorry, that's incorrect. That's 4. Let's see, 4, 10, 4. Okay, so it's this one right here. This is 40 right here. And they would have to rotate this in order to see that. Okay, number 3, more on microscopes. When viewing a prepared slide under the compound microscope, a student has to remove his glasses. This means he will need to readjust for fine focus with which part here. So when we are using a microscope and we want to zoom in, what we do is we raise this stage here. This is the stage. That stage has the specimen on it. That specimen could be like right here. The light passes through and hits that specimen. The light then goes through the objective and then through the ocular, and then you see it, right? So we have to raise the stage to zoom in, and to do that we could use 2 different knobs. This is the coarse focus, and this is the fine focus. So the coarse focus is just bigger, and it's going to create a bigger movement, whereas the fine focus will create a very small movement for very precise changes. So we're using this picture here, so it's a slightly different microscope than what we have. The coarse focus is just the big knob up here, and then the fine focus is the small one, so the answer is 4. Number 4. The picture shows some containers of different foods that were left in the open for 2 days. Which question would be answered by this experiment? So we have a very, very simple experiment. We're just leaving food out. So if it's that simple of an experiment, we can only ask a simple question, really. And this question is an anti-assumption question. You'll see a lot of these on the SOL where they ask you to strictly make an observation and not make any assumptions at all. And so what we have to do is avoid assumptions here. So for example, C would be an assumption, like, well, not really. You could measure it. Let's see. How does a fly digest different foods? Actually, none of these are really assumptions. I don't know if this is an anti-assumption. But this point is relevant. Do we have the correct setup and equipment? So these are questions here that could be asked, but do we have the equipment to actually ask them? So for example, B, which food attracts flies from the greatest distance? Do you see in these materials, do you see a measuring stick? Do you see any way to measure distance of flies or anything like that? No. So that's not the answer. We don't have the equipment. How much energy do the flies get from different foods? Well, how do you measure that? How do you measure the amount of energy? To measure energy, we have to get calories. You have to burn a food. You have to measure the heat coming off of it for the length of time and duration, and that gets you a measurement of what's called kilocalories. You don't even need to know any of that. You just need to know that we don't have any equipment to measure energy. So that's not the answer. And then how does a fly digest different foods? Well, to see that, we would actually have to have equipment that could measure the metabolism of the actual fly, and we don't have that either. All of these are way too complicated a question for a simple experiment like this. This is the only thing that we could actually measure. So we could just sit here and we could watch, and maybe the flies will all come to the orange, a couple will come to the bread, and a few will come to the fish. And so we would say the orange was the best, right? That is a very simple experiment that we could have here. All right, so anyway, moving on. Number 5. This is a simple question. So an experiment was conducted to test the effectiveness of 4 different fertilizers on plant growth. 2 grams of each fertilizer were to be diluted in 9 milliliters of water before adding it to a plant. Which of the following measuring devices would introduce the least error into the measurement of 9 milliliters of water? That is a mouthful, and we can actually just get rid of all of this. The bottom line of the question is right here. Always try to bottom line the question, try to figure out what it's really asking. And what it's really asking is, how can you get the most accurate measurement of 9 milliliters? So here I have some different beakers. This is a 10, this is a 100, and this is a 50, right? So if I want to get 9 milliliters on the 10, all I have to do is go to the line here that has 9. If I want to measure 9 in the 100, I have to first of all figure out what each of these dashes mean. So here's 10 right here. I can't fill to 10 though, I have to fill to 9. So where is 9? Well, each of these dashes is, let's see, 1, 2, 4, 5. Each of these dashes is 1, right? But there's no dashes here. So this is 5, 6, 7, 8, 9. So I have to fill to this little dash right here, right? And that little dash, filling to that precise of a mark, it means there's going to be human error. So the bigger the measuring tool for this small measurement, the more human error there's going to be, because you have to use your eyes to get to that exact spot. Whereas here, you don't have to do that as much because there's a clear place where 9 is marked. So the answer is 10. However, if I was measuring, 50 milliliters of water, the answer would be this one. If I was measuring 70 milliliters of water, I would want to use this one, right? Okay, moving on, 6. According to the graph, how many mice will be born in week 5 if the trend continues? So can you see the trend? The obvious trend is it's going up, but if you look carefully, you can see week 1 is doubled right here. We can actually see that there's twice of week 1. And then here's week 2, and we have doubled it. And then here's week 4. And once again, we are just doubling that. And so if we do this again for week 5, all we have to do is double it. And what is double 80? 80 times 2 is, so this would be 80, and this would be 80, just like this was 40, and this is 40, this was 20, this was 20, this was 10, this was 10, this was 5, or sorry, 10. So the answer is obviously 80 plus 80 is 160. 7. An experiment is designed to clear an oak hickory forest and replace the area with pines. Which of the following species would be the most threatened by this experiment? And so here we have these different birds, and they all live in similar and different areas. So these little lines here represent where they live. We actually don't even have to look at these because we're only looking at oak and we're looking at pine. And so they're saying they're clearing the oak. So this is all going to be gone, and the pine will be, oh sorry, my bad. I meant this. This will all be gone. So which species is the most affected? Well, if you look here, the summer tanager still will have pine trees, the cardinal still will have pine and grass, whereas the hooded warbler is only going to have a small little bit that they can live in. So the hooded warbler, which is actually this guy here, the hooded warbler is going to be the most affected because it doesn't, I mean, it mostly lives in hickory oak, right? Okay, number 8. In the table, which item of data is most likely invalid? So this is just following trends. And so you can see here that you start low with the numbers and then they go up, 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 and then they fall, right? And we can see that same trend with most of these, right? This goes up, 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 and then it falls, right? And then this goes up, 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 and then it falls. So those are all the same, but then we have something different with this line. It goes up, up, way up, down, down, right? And so on June 15th, you can see that the high point was there, whereas most of these have high points here on June the 1st, or sorry, July the 1st. So the weird point is the second plot on June the 15th. Nine. In the lab setup pictured above, or actually to the side here, a student is trying to determine the effect of pollutants on the growth of 3 groups of seeds. The results will not be valid because the experiment has no what? Okay, so we're looking at a couple things. We're looking at effects of pollutants on the growth of 3 groups of seeds. So independent variable is the pollutants, because that is causing an effect. And then the dependent variable is the thing we are measuring, and that is growth. And we actually have multiple seeds that we're testing this with, which is fine. That just basically means 3 experiments. And that's fine. You can have multiple experiments in one experiment, just as long as each one of those experiments only has one independent variable, and that is pollutants. Okay, so what's the problem here? They have motor oil, they have dish soap, and they have antifreeze. What's the problem? The problem is they should have a 4th group, and that 4th group should just be regular soil, because if you're going to measure the effects of pollutants, well, compared to what exactly, right? If, for example, I had an energy drink, and my claim was that it would make me 10 times faster than normal. If I do an experiment where I take the energy drink and I run really fast, that's not good enough. I'd have to also run without the energy drink, right? I mean, what's the use of investigating something if you don't compare it to normal? And so the control group is like normal, like without the thing you're testing, what would you do? So without the pollutants, how would the seeds normally grow, right? So that's called a control group. You have to have a control group to compare with. 10. According to the data in the graph, during which time period did the overall bluegill population decline? So this is just subtraction, really, or just looking at the overall points. So if we look at 1993 and 1996, 1993, each of these is 1 year, so that's 1, 2, 3. This is 1993 to 1996. Here's 1993, and then we go up, and here's 1996, right? So this is just, can you look at a graph? So the start point here is, I know it goes down and then back up, but you just look at the start and the end. This was an increase, and this is looking for decline. So that's not the answer. Then 1999, so let's just do that again. 1999 to 2002. 1999 would be right here, which is a year before 2000, and that point is right there. And then 2002 would be right here, and that's right there. That is also an increase, so that's not the answer. I'll just use a different color. 1990 to 1993, which is 1, 2, 3, right here. So right there, that is also an increase. So then obviously the last one must be the answer. and that is just 1996 to 1999, which is there and there, and there we have it. There's your decrease. Easy peasy. I love those questions. All right, just can you read a graph, basically. Okay, so obviously this is a question about a doge. So a student hypothesizes that thick leg muscles are an inherited trait of doges. The student collects data on several doges, and the data shows that doges that live outdoors have thicker leg muscles than doges that live indoors. What should the student conclude? Okay, so this is really simple. We just don't make any assumptions. Just look at the simple evidence. Like this guy he's a thick boy. He has thick leg muscles. So obviously he exercises, or he's more physically active. Well, which I guess actually is an assumption, but it's the least assumption. So there's something called Occam's razor, which is simply the saying that the simplest explanation for something is most likely true. Occam's razor. Occam was a scientist who came up with that. So what is the simplest explanation for a dog having thick legs? Okay, dogs that inherit thick leg muscles may not survive indoors. So what that means is if you bring a dog home that has thick legs, it's going to die. I don't think so. I don't think that matters. Okay, dogs with thick leg muscles may require more exercise than dogs with thin legs. So again, they're looking at thick leg muscles are inherited dog traits. The students collect data. Okay, so they're just saying that outdoor dogs have thicker leg muscles. So out equals thick, in equals thin, and that's all they're saying. And then what should the students conclude? All right, so dogs have thick leg muscles, require more exercise. So because they need more exercise, there's a difference in survival in the environments. I don't know about that one. That's a complicated answer. All right, inheritance of thick leg muscles may be associated with coat thickness. I don't know where in here it mentioned coats, so we're going to get rid of that. Inheritance alone may not account for thick leg muscles in dogs. So that's probably the answer. It's not just about inheritance. It could also just simply be how much exercise do they get. If they're outside, they're probably moving around a whole lot, so they need thick legs. If they're inside, they're sitting on the couch and they're getting fed every day and they're not exercising. So they probably will have thinner legs. So this is just the simplest answer. That's Occam's razor. It avoids assumptions as much as possible. So it is an assumption, but it's the simplest assumption. It is Occam's razor. So we're just going to go with that one. Okay, another graph question. I love graph questions. You should always celebrate when you see them because the answer is staring you in the face. Okay, the above information was collected in the field while studying the effect of pH on the growth of the duckweed plant. The data shows that duckweed has optimal growth. Optimal growth. What does that mean? It means that that's where they do the best. So straight away, let's look for number of duckweeds, and you can see that 500 is optimal. That's where they're growing the most. And if we just look at pH, that is a pH of A and pond C. So it's asking you, studying the pH, pH of what? So it's asking you what pH gives the best growth, and this is so easy. I love graph questions. Always celebrate when you see them. It couldn't be easier than that. All you have to do is triangulate things, zoom in, and get your answer. All right, so 13. Scientific method. A student's experiment showed that earthworms move away from light. Photosensitivity, I guess. The statement should be classified as what? So, a key word here that we need to look at is showed. It already showed it. We've already done the hypothesis. We've already done an experiment where we shine the light on them. We've already looked at the results, and those results are telling us that that they do move away. So what we have here is just a conclusion. We've already done all of this. We've already made our hypothesis and inferences. We've already made predictions. We are at the conclusion step because the experiment showed. So, pretty straightforward question. All right. A student plans to conduct an experiment to determine how a change in pH of water affects the population growth rate of algae. To ensure her experiment data is valid, she should do what? So here is a pH table, and you can see before 7 is acidic and after 7 is basic, which we also call alkaline. And so we have acidic and alkaline, and the experiment is simple. She's testing different amounts of pH on algae growth. So she'll use like a pH of 4 and then look at the growth. She'll use a pH of 9 and then look at the growth. Look, pH of 12, look at the growth, and try to see a difference, right? So to ensure that her experiment is valid, she should also do what? Should she hold the acidity of the water constant? So if you're holding the acidity of the water constant, you're not testing pH. The goal is to determine the change in pH. So pH is actually what we want to change. It is the independent variable. So you don't want to hold your independent variable constant, or you're not even testing anything, right? So that's not it. So I know that that's an answer that you probably want to straight away because you saw this and you thought, ooh, that's often the answer. In this case, it's not. You don't want your constant to be your independent variable. That's the thing you're actually testing, and this is just not even testing it. All right, so vary the temperature of the water used. So that is introducing another independent variable. Our independent variable is already pH. That is what we're testing. We don't want to also test temperature, so that's not the answer, because then we don't know what the effect is. We don't know if the algae here is growing because of the temperature or the pH. All right, C, control the final algae population sizes. What? The algae population is the dependent variable. That's what we measure. We don't want to control that. We don't want to change that. That would be like me testing the effect of fertilizer on grass growth and then just cutting the grass before I measured it. You know? Like, what would be the point of that? What would be the point of changing the results of an experiment before measuring those results? That's ridiculous. So no. Finally, use the same species of algae in all trials. Yes, because if we used a different species in each of these trials with these different pHs, that's another independent variable, just like the temperature was another independent variable. So we want to use the same algae in all of them because different algae could grow differently in different pHs, and that's going to create too complicated a result. We're not going to be able to figure out what actually caused the algae to grow. So this is a very simple question. It's just quizzing your ability to understand that you only have one independent variable and everything else must be constant, except for obviously the independent variable itself. That is not held constant. All right. Students in biology class thought that salt water would affect the movement of planaria. They put 25 planaria in salt water and recorded their observations. What is the flaw? Okay, so they're saying that the planaria swims slower in salt water. That's a fine hypothesis. So they're just taking salt water and they're just dropping the planaria in there and they're watching it move. That's great, but compared to what? For this to actually work, they would need a second class with just regular water, and they would need to compare. But they're not doing that. They're just throwing it in salt water. So the fresh water would lack the salt, and the salt here is the independent variable, and so what they're lacking here is a control group. This would be the experimental group, and this would be the control group. Sorry for my handwriting. So there you go. It's A. Okay, number 16, almost done. Which set of materials should be best, or would be best, to look at onion skin under a microscope? Okay, so onion skin is challenging because just like your cells, if you were to cut that very thin, you might imagine that it's actually transparent. Light just moves right through it. And so we wouldn't be able to actually see it. Light has to bounce off of things for us to see it. So what we actually have to do with cells like onions or human skin cells is we have to actually stain them. So these are the materials. We're going to go ahead and get rid of these answers because there's no stain. We need stain if you want to see something transparent. Okay, so then the next thing is when we put a slide down, and I'm just going to use a different color so you can see it. If we put a slide down here, that slide can only be so thick. And so we need another little tiny cover slip, and then we put our object in between the cover slip and the slide. And so we need that cover slip to be very thin. And the reason why is because if we tried to use 2 slides like this, as you can see, there would be no room. Also, the light would just not really penetrate both of those slides very well, and you wouldn't get a good image. And so it's always better to use a thin cover slip than using 2 slides, and so the answer is option 4, not 2. 17. These 3 samples from the same live culture, so that's just living bacteria, were all viewed at the same magnification. Which conclusion is best supported by the observation? So we have a bunch of bacteria and sample A, and who cares? It's under 400x, who cares? There's a lot of extra information here that we don't really even need. So then we add some sort of chemical with a little dropper there, and then we wait a couple days, and then we measure it the next day, and we look under the microscope, and now look at the sample B. So from sample A to sample B, a whole lot of stuff is dead. Whatever was in this little dropper killed them, but it did not kill all of them, did it? There are still some leftover bacteria in here. And so what must be different about those bacteria? If we compare the bacteria in sample A to sample B, what's the difference? Well, obviously, the stuff in sample B was the leftover bacteria that could not be killed. And so then they came back and they grew. And so what happened here? So what happened was the bacteria here are immune to this chemical, whereas a lot of the stuff here in sample A was not. Okay, so the sampling technique used did not produce accurate data. Okay, so they're trying to trick you here by saying that the goal was to kill everything, but it's not. It was just to observe the effect of the chemical. So the sampling technique did produce data, and we can see that here. The bacteria culture was unaffected by the chemical agent. Well, obviously it was affected. Look at the difference between sample A and sample B. There was definitely an effect. The culture became contaminated by airborne bacteria. That is an assumption that we cannot make by looking at this. Avoid assumptions. Okay, so the culture included some bacteria that were resistant. There it is. So there were some of these in sample A that were resistant, and they survived. Hold on, there's somebody at the door. Sorry about that. Where was I? There was someone at the door. Oh, right. So sample A had some stuff in it that was resistant, and they are the ones that survived. The rest were killed. And so the ones that survived were resistant. We actually have a lot of problems with this. Bacteria tend to become resistant to the drugs that we use. So this was probably an antibiotic, and it killed what it could, but it couldn't kill them all. And we actually have that problem with treating diseases these days. Okay, moving on. So a scientist develops a hypothesis, designs an experiment, and obtains data that supports her hypothesis. I like that they used her there. It's so typical. They always say he. Very cool. All right, so which of the following best describes when a hypothesis becomes a theory? So what is a theory? That's what this is asking you. And so here's a quick video about all of these words you've probably heard before in science. I think this guy says it better than I do, so we're just going to watch him.
We need to get something straight. Evolution by natural selection is a theory. So is climate change. But people keep saying that like it's a bad thing. Neatly in our brains. But what is truth? Are there different levels of truth? Are some truths truthier than others? Well, I don't know, but I do know this. Science is the absolute best tool we have for understanding how the universe works. And theory is not a four-letter word. If we're going to trust science together, the least we can do is speak the same language. Words like fact, theory, hypothesis, and law mean something totally different to a scientist than the way they're used in everyday speech. So let's get them straight. Facts are really just observations about the world around us. And we observe things every day, like that it's bright outside when I look out the window. And we often develop explanations for those observations, like, okay, the sun is probably up. Congrats, we just developed a hypothesis. But a hypothesis isn't something you prove. It's something you test. So let's walk outside. It's bright, the sun is up, hypothesis confirmed. Way to go, we did a science. We often come up with multiple hypotheses to explain an observation. We just eliminate the ones that are wrong. What's left over is not a theory or a law or an ultimate truth. It's just a possible explanation for something, one that can lead us to new hypotheses, which may agree or disagree with the original one. It's a never-ending story, only without the big fluffy dragons.
That's so good.
When enough hypotheses have gotten the old scientific check mark, we can pile these all up and turn them into something greater, a theory. A theory is the way we know something works.
Okay, so let me just summarize because this is going to get a little bit long-winded here. We've got hypothesis, 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 right? That is all used to create an overall theory. So a theory is based on many hypotheses coming together, sort of like bricks in a foundation. And then that becomes a theory. And then that theory is used as a foundation for other hypotheses. So for example, there is the theory of evolution. That theory was built upon Charles Darwin's hypotheses, like for example, that nature was difficult, that there's selective processes in nature, that there is a survival of the fittest, that nature prunes out the weak. These were all individual hypotheses that were put together to sort of create the theory of evolution. Now that theory is a foundation upon which we can now build modern hypotheses on. So for example, a current hypothesis that we can use to build on top of that would be the hypothesis that we are related to chimps, that they are our closest ancestor. That hypothesis is only possible because of the theory of evolution. And so a theory is just a collection of hypotheses that support more hypotheses. So it's a bigger deal is what I'm trying to say. So then back to the question. When do you actually have a theory? So is it when it is supported by consistent data from many experimental trials? Well, that would be helpful, but let's keep reading. When the official scientific method is followed. So that just means that you've got correct science, but that does not give you a theory. A theory is multiple hypotheses together. So when a website, no. Just because a website says it, we all know that there's a lot of BS out there on the web, and you need to be careful of it. So when one good set of scientific data supports a theory. No, you need more than just one set. You need a lot of information. So the best answer is A, when it is supported by consistent data from many, many trials. 19, home stretch. A student wanted to compare the transpiration rates, so that's just how quickly you release water from your body. Transpiration rates of two different plant species at varying levels of humidity. So we have a independent variable, and that is humidity. I always like to write this down as I go. And then the dependent variable is going to be the transpiration. So quite simply, what they're changing here is humidity of the room, and they're looking at the transpiration of the plant. So each plant group, the results are recorded in the data table shown. Each plant group was given the same amount of water and light throughout the investigation. Which procedure should have been performed to prevent error? So we're looking for error. I love these questions because they're always easy to me. That's just me. So here's sort of the experiment, and we're trying to figure out what's wrong. So let's see. The humidity is the independent variable, and you can see that we varied that here, and that's good. That is what you are supposed to do. You are supposed to vary your independent variable. So this is what we're actually testing, so we do want to have our independent variable, humidity, changing. So that's good. We can check that off. That's fine. Next, we have different plant species. So are they testing for that? Let's go back real quick. Are they testing different plant species? Two different plant species. Okay, that's fine, I guess. So we have two different plant species, and we're testing differing humidities on both. I suppose that's fine. That's really just two experiments being done at once. But then we have sand and clay. So now it's getting confusing, right? Because we have different mediums too. We have sand and we have clay. So we're getting a lot of independent variables here. And then the dependent variable is the transpiration. So nowhere in this did they mention sand or soil. Varying levels of humidity. So they want humidity to change, and they want to look at different plant species, and they want to look at transpiration. Nowhere in this question did it mention anything about soil or sand. So what should they do? One type of plant? No. They wanted to test different plant species. The percent of humidity should be kept the same? No. Humidity is actually the main thing they're testing. Both groups should have been tested in the same soil. There it is. There's nothing in here that mentions soil type, and yet they varied the soil type. So that's the answer. And then the last one says fewer plants should have been included. No. The more times you do an experiment, the more valid it probably is when you get your result. The more test subjects you use, the more trials you have, the better. It's sort of like, if I say that when you flip a coin, there's a 50-50 chance of getting heads or tails. If I do an experiment to test that, but I only do it 2 times, I could easily get heads twice and conclude that I was wrong. But if I flip 100 coins, chances are I'm going to get closer and closer and closer to a 50-50 chance. So the more times that you do an experiment, the more likely you are to remove statistical anomalies, if that makes any sense. Okay, so last question. Thanks for hanging in there. This is a long one, but it kind of has to be. I mean, it's 20 questions, and you need to at least spend 3 minutes on each, so do the math. All right, which trial in the table of data shown has questionable data? So once again, I love these questions. You just look for the weird data. So this goes 3, 6, 10, 14, 5, 7, 10, 14, 2, 4, 9, 12, 4, 7, 17, 13. So you can see here, this is the weird one. It's going up and then down, whereas all of the rest of these just go up the whole way. This goes up, this goes up, this goes up, and then this goes up and then down. So it's going to be Z, that's the weird one. That's just looking at trends, and there you have it. Sol Strand 1. Hope it wasn't too hard. I'll see you in the next one.